So today we are going to discuss about the mitochondrial genome. As you know, the mitochondria is present in the cytoplasm of the cell. It is an organelle and it is semi-autonomous in nature. And why it is semi-autonomous in nature? Because it has its DNA and proteins, but it is not completely independent. It is dependent on the proteins which are coded by the nuclear genome. So that there is, because DNA needs to be transcribed to RNA. For that, they need the proteins. For the replication of DNA, they need the proteins. From where these proteins are coming? These are coming from the nuclear genome. That is why it is not completely independent, but it's a sort of partially independent, and that is why it is known as the semi-autonomous organelle. So mitochondrial DNA, the very important thing is this is circular in nature and it has 16,568 base pair. It carries the genetic information for 2 ribosomal RNA, 22 transfer RNA and 13 proteins. So what are these proteins? You have read about the electron transport chain, which is formed by the complex 1, 2, 3 and 4. And when it is mixed with the complex 5, which is the ATP synthase, this whole complex system is known as the oxidative phosphorylation, where there is a production of the ATP molecule. So what are these complexes? These are the complexes, again, these are the proteins. So you can see the 13 proteins which are coded by the mitochondrial genome are involved in the oxidative phosphorylation. For example, 7 of these proteins are forming complex 1, 1 cytochrome B is forming complex 3, 3 is forming complex 4, and 2 is forming the complex 5. If you see here, you can see mitochondrial DNA is circular in nature. These are the proteins 1, 2, 3, 4, 4L, 5, and 6. So total 7 proteins coding for the complex 1. This is complex 3, cytochrome B, then COX1, COX2, COX3. They are forming the complex 4. And you can see the A6 and A8. This is forming the complex 5. So that is what is it. It is written ATPase6 and ATPase8 of complex 5. Again, if you see here, for the conversion of this mitochondrial DNA to proteins, they need the nuclear proteins. And nuclear genes encode about 70 additional proteins that make up the OXPO system and about 1,000 other proteins that promote the replication, transcription, and translation of the mitochondrial genome. And that is why it is semi-autonomous. It is not completely independent. Now, if you see the maternal inheritance. So this is, let's say, the oocyte. So you know this is the oocyte, let's say, and this is your, let's say, sperm, and this is the neck of the sperm. This is the head of the sperm, right? And this is the tail. And if we talk about this is the oocyte. So whenever there is a fusion of a sperm with the oocyte, this neck portion is not fusing with the oocyte. And this is the region that contains the maximum mitochondria. And because of this, the mitochondria of the zygote, whenever there is a fusion of the sperm with oocyte, that is forming the zygote. Now, because this neck is not fusing with this oocyte, the mitochondria which is present in the zygote is coming from only female most of the time. So that is what it is mentioned here. If you see here, you can see the mitochondria of the zygote come from the oocyte, that is from the mother, and almost never from the sperm, that is from the father, and this is form of transmission is called the maternal inheritance. You can see here in this pedigree, this is the female, you can see two of its kids are affected by the disease. But now this male is affected, but the offspring of this male is not having any disease. It means what it means. And here you can see this is the female. Because it is affected, it is having affected children. Again, this is the female affected, and this is why this is having the affected children. So it means, so if I write here, this is the affected female. This is the affected male. Can you see any of the children affected? You can see offsprings are not affected. So what it means? It means this is the example of maternal inheritance. Because if the female is affected, then only the disease is transmitted because it is going via mitochondria. Okay. Then you can see the human oocyte has almost 0.1 million of mitochondria, but it is reduced to in between the range of 10 to 100, and this kind of process is known as the bottleneck process. The process of reducing the number of mitochondria from 0.1 million to less than 100 is known as a genetic bottleneck. So if you see here, you can, why it is known as the bottleneck? Because this is 1000 mitochondria, now it is reducing to 10 or 10 to 100. Here it is 0.1 million. And that is why this is the process known as the bottleneck, which is like a bottleneck. If any mitochondria, if uh, the mitochondria, if it has the gene which is mutated gene, and if it survives, then definitely that cell is going to have that particular mutation. So now the term homoplasmy means whenever the mitochondrial genome is having either the wild type sequence or the mutated sequence. But in case of the heteroplasmy, it contains both mutant and the wild type sequence. So here you can see, suppose this is your zygote and or the cell, and in this the mitochondria, and the mitochondrial gene is responsible for the production of ATP. Now this gene is mutated, let's say, then there will be less or reduced production of the ATP. Now if this particular cell is belonging to your heart or cardiovascular system, what will happen? Or if it is your nerve cells, because they need more energy, in that case what will happen? Because there is less production of ATP, so this cell is going to suffer. But if it is like the cell which is not requiring any high energy, for example RBC, then the effect is going to be less. So this is what it is mentioned here. If the mitochondrial gene mutation reduces the production of ATP, then the cell that require a high energy, like your heart muscle cells, then in that case that is going to be seriously damaged. But if it is the cell, it is requiring a very less energy, then the effect is not going to be very high. In the heteroplasmic cell, that contains both mutant and the wild type mitochondrial genome. So whether the cell is affected more or not, it will depend on the proportion of mutant to wild type. If mutant is more, then it is going to be affected. If it is less, it is going to be less affected. Like that. So that's all about today's lecture. Thank you.